membrane bound enzymes are present. If you take the function, normally we say lipid synthesis. Lipid synthesis function is there for smooth endoplasmic reticulum, lipid synthesis. Here they are broadening it. What are the lipids pointed to you? What are the lipids pointed to you?
Oils, steroids.
And phospholipids. Second thing, they say metabolism of carbohydrates. Carbohydrates are now, according to my references, certain disaccharides are synthesized by the smooth endoplasmic reticulum. Certain disaccharides are synthesized by smooth endoplasmic reticulum. This function producing transport vesicles, okay, is already we studied in the rough endoplasmic reticulum too. Rough endoplasmic reticulum produces transport vesicles. Smooth endoplasmic reticulum also produces transport vesicles. Fourth function, very important function. What is the function? Read this word. Read this word.
Detoxification.
Detoxification. Detoxification means breaking the toxins. Understood?
Yes, sir.
For a simple example, now say you are taking 2 Panadols inside your body. 2 Panadols mean 1000 milligram paracetamol. 1000 milligram, what is it? Paracetamol. This paracetamol will be taken into our body. In this, about 90% will be broken by our liver. Rest only will give you the relief. The relief only 100 milligram will do that. 900 milligram will be broken. That is called detoxification. Detoxification is there in our body for our protection. Okay, if any medicine, any toxin enters our body, smooth endoplasmic reticulum will be breaking it. Okay. Last function, storing which ions? Storing which ions?
Calcium.
Calcium ion. Okay. This is mainly, it is mainly, not only, mainly in the muscle cells. Which cells?
Muscle cells.
Mainly in the muscle cells, because muscle contraction requires calcium ions. So it is mainly in the muscle cells. Please mark it down.
Done, sir.
Right. So, are you clear regarding the smooth endoplasmic reticulum? Are you clear regarding the smooth endoplasmic reticulum?
Yes, sir.
Yes. So, with that one, smooth endoplasmic reticulum is over. We are coming to the heading called, what is it?
Golgi apparatus.
Golgi apparatus. Okay. If you take Golgi apparatus, okay, it is another unit of endomembrane system. Last class, I think I introduced you the endomembrane system. Correct?
Yes.
Did I introduce you the endomembrane system? My children. Did I introduce you the endomembrane system? Yes? Yes, no?
Yes, sir.
Yes. So this is another component of endomembrane system. In the Golgi apparatus, okay, if you look at this diagram, you can see several flattened units are stacked one on the other. Can you see that one? Several flattened units are stacked one on the other. One, another one above, another one above, like that. Several flattened units are stacked one on the other. Can you see?
Yes, sir.
Yes. These flattened units are called cisternae. What is the name given? What is the name given?
Cisternae.
Cisternae. In this picture, can you see the word cisternae? Here.
Yes.
Yes. So here, this is the cisternae. This is the cisternae. This is the cisternae. This is the cisternae. There are many. What is it? Cisternae there. Clear?
Clear, sir.
Right. In the Golgi apparatus, there are two phases. This phase is called cis phase. This phase is called trans phase. How many phases are there? How many phases are there? Two phases are there. One it is cis phase, other one is
Trans.
trans phase. If you take the cis phase will be situated closer to the endoplasmic reticulum. Cis phase will be located closer to the endoplasmic reticulum. From the endoplasmic reticulum, vesicles will come and join the cis phase. The secretions in those vesicles will be added to the Golgi apparatus. Understood? Understood.
Yes, sir.
Cis phase is situated closer to
Rough endoplasmic reticulum.
rough. Did I tell rough? Did I tell the word rough? Ama. I told a common wording. Situated closer to the endoplasmic reticulum. What is the benefit? From the endoplasmic reticulum, secretions are packaged in vesicles and sent to the whom? From endoplasmic reticulum, they are packaged and sent to whom?
Golgi apparatus.
Golgi apparatus. Which phase of the Golgi apparatus will receive it?
Cis phase.
Cis phase. Cis is a receiving phase. This is a receiving phase. Thereafter, if you go to the trans phase, from the trans phase, new vesicles will be formed. Okay, so these vesicles are made from the Golgi apparatus. They are the vesicles of Golgi apparatus. Clear? They are the vesicles of whom?
Golgi apparatus.
Golgi apparatus. Okay, they are budding off from the trans phase. They will be carried to other parts of the cell. So, which is the receiving phase here?
Cis phase.
Cis phase. Which is the shipping phase here?
Trans phase.
Trans phase. Are you clear?
Clear, sir.
Here also the inner space is called by the name lumen. Lumen, last class I told you. Lumen is the inner space of the endomembrane system, internal membranes. Okay. Are you clear regarding the Golgi apparatus? Are
Yes,
you
sir.
clear regarding? Okay. So what kind of units they are? Flattened sacs. Flattened sacs are stacking one on the other, one on the other, one on the other. Every flattened sac is called by the name cisternae. There are two phases. One we call cis phase, one we call trans phase. Cis phase is located closer to ER to receive the vesicles from ER. Trans phase is budding up vesicles. Those vesicles are called by the name. What kind of vesicles?
Vesicle.
Secretory vesicles. Okay. Golgi complex is abundant in which kind of cells?
Secretory cells.
Our salivary gland cell. Okay, tear gland cell, sweat gland, secreting places. Secreting places. Clear? Secreting places have, what is it? Lots of Golgi. Secreting cells have lots of Golgi complex. Right. What are the functions of Golgi? One function, collecting, packaging, and distribution of material. Mainly they collect from ER. Mainly from whom they collect? From whom they collect?
ER.
ER. Second function, manufacturing. Manufacturing. Wait. Manufacturing. What is manufactured?
Cellulose and
Cellulose and
And
non-cellulose cell wall component. Remember, okay, in this one who are included? Hemicellulose, pectin-like materials are included. So cell wall components are synthesized by the Golgi. Final function.
produce lysozymes.
Producing lysosome. Are you clear? Are you clear?
clear.
Good. Now, regarding whom? Lysosomes. In the case of lysosomes are vesicles, single membrane bounded, what is it?
Vesicles.
Vesicles. Those vesicles are taking part in which activity? Which activity? Digestive activity. Okay, read this name.
Hydrolytic enzymes.
Hydrolytic enzymes. It is the digestive enzymes. In your O level, you have studied several digestive enzymes, something like lipase, correct? Amylase, trypsin, pepsin, right? Those kind of enzymes. Whatever the digestion taking place inside a cell, who will take part? Who will take part?
Enzymes.
Lysosomes will take part. Okay. In that case, lysosomes have many functions. One of the function is this. What is this word? Can you read this word?
Phagocytosis.
Phagocytosis. My children, phago mean eating. Phagocytosis means cell eating. Understood?
Yes.
Okay. In the cell eating, who will take part? Lysosome will take part. Okay. Now look at this picture. Can you see a bacteria there?
Yes.
Yes. this is, okay, say it is an amoeba. It is an, what is it? Amoeba. Amoeba, okay, you can see amoeba catch the bacteria and take it inside. Right. After engulfing the bacteria, now bacteria is inside the amoeba. Can you see? Can you see?
Yes, sir.
Yes. Now this unit is joined by whom?
Lysosome.
Lysosome. Lysosome will add enzymes to the bacteria. Because of that, bacteria's body will be digested. Bacteria's body will be, what is it?
Digested.
Digested. From that one, the amoeba will absorb whatever essential for it. Understood? Whatever essential for it, it will absorb. After absorbing, rest is there, no? Rest is there, no? Okay. It will be removed out of the cell. That removing out of the cell is called by the name
Exocytosis.
exocytosis. So two words, one it is phagocytosis, other one is, what is it? Exocytosis. Are you clear? Are you clear?
Yes, sir.
In the point of view of an amoeba, this is nutrition. Bacteria is the food for it. This same thing can happen in our body when white blood cells are destroying bacteria. Understood? Understood?
Yeah.
Yes.
When white blood cells are destroying bacteria, also the same thing can happen in our body. When you go in the biology, you will see in several places this kind of an activity is going on. So here, my children, there are two words to catch. One it is phagocytosis, other one is, what is it?
Exocytosis.
Exocytosis. Taking the bacteria into the cell. digesting with the lysosome, phagocytosis work, then sending the unwanted materials out of the cell. That is
Exocytosis.
exocytosis. So, if you take the tube, in the tube, you children are given first thing home. First thing home. Read the first one.
Largest food particle received by phagocytosis.
It will be digesting the food particles by phagocytosis. Food particles, as I told you, in point of view of an amoeba, bacteria is a
Food particle.
food particle. Clear?
Okay.
Are you clear?
Yes, sir.
Good. Second one, they speak about which process? Exocytosis. Correct? Who are sent out by exocytosis?
Residue material.
Residue material, unwanted material. Clear? Clear?
Clear, sir.
Good. What is the third function? Third function. What is the function?
Digest worn out organelles.
Digesting worn out organelles. Now, when a cell is functioning, the organelles there will be becoming little by little older, less effective. Understood?
Yes, sir.
And that is common now. Now, if you buy a machine, when time goes up, what would happen? It will be becoming weaker.
One.
Correct? One stage will come. This organelle is no more beneficial. Correct?
Yes, sir.
In that time, what the cell does is, all those kind of organelles are surrounded by a membrane. All those types of organelles are surrounded by a membrane. Into that membrane, lysosome will be adding its enzyme. Due to those enzymes, those organelles will be digested. They are removed from the cell environment. Clear? They are removed from the cell environment. They are not essential. They are not beneficiary. Clear? Are you clear?
Clear, sir.
Good. And finally, okay, remember, this third one has a unique name called autophagy. What is the name? What is the name? Autophagy. digesting worn out organelles is called, what is it?
Autophagy.
Autophagy. Our resource book doesn't include that word, but it is okay. Better you know. At least it will be helping you in a past paper question. Clear?
Clear, sir.
Okay, don't take any past paper questions or anything. Okay, all the relevant materials I will be providing you, use them. Okay,
Okay, sir.
okay, right. There are reasons for that. Now, if you take the past paper, you know your syllabus, new syllabus has started in 2000, what is it? 19. 2019 is the first A-level batch of new syllabus. Then 2020 children did the A-level, 2021 have done the A-level. Okay, 22 is going to do the A-level. So how many years past papers we have according to the new syllabus?
3.
3 years only. All the questions beyond that, 18, 17, 16, 15, 14, all are old syllabus. But from those old syllabus, we can pick worthy questions. Certain questions will be there, we have to modify. We have to modify according to the new syllabus. So if you buy 10 years past paper, in that 10 years past paper, only 3 years papers will be relevant to the new syllabus. So what I do is I take those questions, modify according to the new syllabus and have a set of it. When you finish the first second unit, I will give that set of questions in your hands. Then you can continuously practice it, continuously do it. Understood? Okay, like this for every unit, inshallah, I will hand over MCQ, structure, essay, compilation. That's why I told don't take past papers. Okay, I will give you. Clear?
Yes, sir.
Good. What is the fourth function, my children? What is the fourth function? What is the word they are applying?
Autolysis.
Autolysis. Here, autolysis. Auto, what is it? Lysis by lysosome. Autolysis by lysosome. What's happening? It is a cell death. What is happening? What is happening?
Cell death.
Cell death. What's happening? It is in the cell, there will be many lysosomes, no? Correct? Yes or no? Yes or no? There will be many lysosomes there, hundreds of lysosomes there. All those lysosomes will blast at once. What will happen? All the lysosomes will blast at once. Because of that rupture, the total cell will be destroyed. The total cell will be
Destroyed.
This is how a frog loses its tail. Okay, baby frogs can be seen being with
Tail.
tails. When it is turning to adult, the trail will be becoming shorter, finally disappear. What's happening it is, the cells are dying. By which process? By which process?
Autolysis.
Autolysis. One side, this benefit mean, another benefit it is, if any bacteria comes inside a cell, if any virus comes inside a cell, to destroy that virus, this kind of a procedure is there. In this place, the entire cell dies, correct? Am I right? Am I right?
Yes, sir.
All the lysosomes die. In this case, lysosomes are called by the names. What is the name given? What is the name given?
Suicidal bags.
Suicidal bags. Because they are killing themselves and killing the entire cell. So they are called suicidal bags. Understood?
Yes.
Good. Please mark it down. Suicidal bags. Have you completed those short questions on carbohydrates?
Yes, sir.
Yeah. Any doubts there? No. All are clear.
Yes.
Do you have any doubts to ask at me in those short questions?
No, sir.
No, everything is clear. Okay. Right. Thereafter, what is the heading? Read the heading. What type of vesicles? Peroxisomes. They are single membrane bounded vesicles having what type of enzymes?
Oxidizing enzyme.
Oxidizing enzymes. Hydrolytic enzymes are present mean? Hydrolytic enzymes are present mean?
Lysosome.
Lysosome. Oxidizing enzymes are present mean?
Peroxisome.
Peroxisome. Clear? Are
Yes, sir.
you clear? Right. Second thing, it is easy. Peroxisomes can be found in both plant cells as well in animal cells. Third one, peroxisomes are catalyzing the breakdown of whom, what is marked? H2O2. What is that H2O2? Hydrogen peroxide. What is H2O2? What is
Hydrogen peroxide.
H2O2? Hydrogen peroxide. Understood?
Yes, sir.
Yes, hydrogen peroxide. Remember, hydrogen peroxide is toxic to cells. Hydrogen peroxide is, what is it?
Toxic to
Toxic
cells.
to cells. Because hydrogen peroxide is toxic to cells, it is broken into H2O and O2. To which compounds it is broken?
H2O plus
H2O and O2. This breaking down need enzymes. Those enzymes are present in the peroxisomes. Are you clear?
Clear, sir.
So hydrogen peroxide, my children, naturally appear in cells. That is one thing. Hydrogen peroxide naturally appear in cells. Same time, hydrogen peroxide is toxic to cells. So it should be broken to H2O and what is it?
O2.
O2. That needs enzymes. Those enzymes are stored in the
Peroxisome.
peroxisome. Are you clear?
Clear, sir.
Clear. Right. Now, peroxisomes, what are the functions? One, they say like this. What is the function?
Detoxification of peroxide.
Peroxide. That you can understand. Toxic hydrogen peroxide is broken to harmless water and oxygen. Correct?
Correct.
Correct. Second function, photorespiration in plants. When you study photosynthesis, inshallah, you will study the process called photorespiration. Okay? It is happening only in certain plants. Okay? In that one, a toxic material called this name. What is the name? Read this name.
Phosphoglycolate.
Phosphoglycolate. It is also a toxic compound. Understood? It is also toxic to cells. It is also broken down. But this toxic compound is produced only in some plant cells. Peroxide is not so. Peroxide is produced in the plant cells as well in the
Animals.
animal cells. But peroxisome is not, sorry, phosphoglycolate is not so. Phosphoglycolate is produced only in some plant cells. Clear? Are you clear?
Yes.
Right. In the plants, another kind of peroxisome is there. What is the name of that peroxisome?
Glyosomes.
Glyoxisome. This glyoxisome can be found in which tissues of the plant?
Fat storing tissue.
Fat storing tissues. What is the function of it in the fat storing tissues? Converting
Fatty acid into
fatty
sugar.
acids to
Sugar.
sugars. Clear? Are you clear?
Clear, sir.
Clear. Right. So, finishing peroxisomes, we are coming to the next organelle. What is it?
Mitochondria.
Mitochondria. Remember, this type of pictures are there, no? Okay, this right side picture, they are
Yes.
transmission electron microscope picture. What kind of picture? TEM. Okay, TEM diagram of these organelles. Keep in the mind, these pictures you should be able to identify. They will give you these types of pictures in the exam paper and will ask you to identify them. Understood? Understood.
Understood.
Good. So, coming to the detailed idea of the mitochondria. Mitochondria will be a long structure like a sausage. It's an elongated structure. Okay, it is an elongated structure, long structure. You can see in the electron micrograph also, it is a long structure. In this mitochondria, there are two membranes. How many membranes? How many membranes? Two membranes. One we call outer membrane, one we call, what is it?
Inner membrane.
Inner membrane. Between outer membrane and inner membrane, there is a space. What is the name given to that space?
Crystal.
Yes.
No, Baba. Between inner membrane and outer membrane, there is a space. That space is called by the name intermembrane space. Clear?
Clear, sir.
Clear. If you take the inner membrane is folded in this manner. Can you see? This way, who is folded? Which membrane is folded?
Inner membrane.
Inner membrane is folded. These folds are called by the name cristae. Understood?
Yes, sir.
Right. If you go and observe a cristae closely, in the cristae, there will be several stoked particles. What kind of particles?
Stop particle.
Stoked particles. Draw a small diagram and mark the, what is it? Stoked particles in the cristae. STALKED. STALKED. Stoked particle. Have you
Done.
marked it?
Yeah, sir.
Yes. Inside the mitochondria, in the middle of the mitochondria, there will be a semi-solid region called, what is it?
Matrix.
Matrix. In this matrix, you will notice a circular DNA. Could you please draw a DNA? Circular mean not a circle. Okay, you know the meaning. I have already taught you the circular DNA. The DNA like this. Could you please draw one and mark it as circular DNA in your diagram?
Okay, sir. Done, sir.
Okay. Further, in the matrix, there will be 70S ribosomes will be there. Mark the 70S ribosome. Marked it?
Yes.
Good. So, what we described here are the ones given in this note. Could you please read and see the note?
Okay, sir.
Only one thing, two components we have not painted in the matrix. That is hard to paint in the diagram, but is in the note, phosphate granules and enzymes. Others are okay. Read and see first. These ones, read and see. Okay. Okay.
Okay.
Right. Remember, when you study cellular respiration, you will come to you in the unit number 2. In the cellular respiration, there are 4 steps you will study. Glycolysis is one step. Pyruvate oxidation is another step. Krebs cycle is another step. Okay, and oxidative phosphorylation. And electron transport chain is another one. What are the steps of cellular respiration? What are the
Glycolysis,
steps of cellular respiration? Read it.
glycolysis,
Hmm.
pyruvate oxidation,
Hmm.
Krebs cycle,
Hmm.
oxidative phosphorylation, and
And
electron
right.
transport chain.
In this one, glycolysis occurs in the cytoplasm. Rest is there no. What are they? Pyruvate oxidation, Krebs cycle, oxidative phosphorylation, and electron transport chain. They are happening in the mitochondria. Clear?
Yes, sir.
Right. In this one, if you take this one, pyruvate oxidation and Krebs cycle are occurring in the mitochondrial, what is it?
Matrix.
Matrix. Due to that reason, the enzymes relevant to them are present in the mitochondrial matrix. These ones are happening in the cristae of mitochondria. Understood? So those enzymes, the enzymes relevant to oxidative phosphorylation and electron transport chain are present in the state of mitochondria. Clear?
Clear, sir.
Right. Could you please mark this one and keep? Colon. Hello. Okay, completed.
Yes, sir.
Right. So, if you consider the function of it, one function synthesizing ATP in aerobic respiration. Second one, involving in, what is it?
Photorespiration.
Respiration. Right. Thereafter, the heading it is, what is it?
Chloroplast.
Chloroplast. If you take the chloroplast, the right side picture, label it as TEM diagram. This is how, under transmission electron microscope, it will appear. Okay,
Okay, sir.
right. So in this case, my children, here also, how many membranes are present?
Two membranes.
Two membranes are present. Inner membrane is present. Outer membrane is present. Here also there is an intermembrane space. Here also there is a jelly medium called. Only difference is the name of the jelly medium is called
Stroma.
stroma. The name of the jelly medium is called, what is it? Stroma. Okay. In that jelly medium, 70S ribosomes, circular DNA, all what you see in the mitochondria can be seen. But this is matrix. No, this is not matrix, this is stroma. Okay. Here the speciality is inside the inside the stroma there is another membrane system. What is the membrane system? It is called thylakoid system. What is it? What is it?
Thylakoid system.
Thylakoid system. If you take this thylakoid system, it is the one majorly responsible for the absorption of light and initiating the photosynthesis function. It is the one green in color. It is the one, what is it?
Green in color.
Green in color. Chloroplast is green in color, means thylakoid system is green in color. Because thylakoid system is green in color, chloroplast is green in color. Clear? Are you clear?
Yes.
Good.
Yes.
In the thylakoid system, if you go near, okay, in this one you can see there are several button like structures, correct?
Yes.
Every button is called by the name thylakoid. Every button is called by the name, what is it?
Thylakoid.
Thylakoid. When several thylakoids are stacking on the other, the full unit is called by the name granum. Here in this picture, how many granum you can notice? One granum. One granum has many thylakoids. Understood? Understood? Yes. If now I am taking the picture like this, this is a granum. Agree?
Yes, sir.
This is another granum. Agree?
Yes, sir.
Yes. Between the granum and granum, can you see this kind of a unit?
Yeah.
Yes or no?
Yes, sir.
Yeah.
Yes. It should be labeled, my children. It is intergranal lamella. What is the name?
Intergranal lamella.
Intergranal lamella. Have you labeled it?
Yes, sir.
Yes. So, further, if you take the thylakoid membrane, okay, thylakoid membrane structure, right? Thylakoid membrane structure. you will later study this one in full detail. I just take this one. Okay. In this thylakoid, I am showing the thylakoid's detail. Understood? I am showing the, what is it?
Thylakoid.
Thylakoid's detail. In the thylakoid, this kind of units will be present. They are called by the name photosystems. What is the name given?
Photosystems.
Photosystems. Photosystems are the places having the photosynthetic pigments. Photosystems are the places having the, what is it? Photosynthetic
Pigments.
Later, you will study a full detail about the photosystem. But at the moment, just cell biology, no need to go further deeper into the photosystem. Clear?
Clear, sir.
Right. So please read and see this note. Okay. The components present in the stroma are given here. Starch granules, lipid droplets, many enzymes like additional components are also mentioned to you. Other things will be okay. Please go through it. If you are clear, we will further move.
Clear, sir.
Clear. So function wise, it is photosynthesis only. Okay, nothing more. Right. Thereafter, we are coming to the next heading. What is the heading?
Cytoskeleton.
Cytoskeleton. It is a unit found in the cytoplasm of eukaryotic cells made of protein tubules and protein filaments. Okay, where will you see this one? Inside the cytoplasm. Inside the, what is it? Cytoplasm. Right. It is called by the name cytoskeleton. I want to catch the word here. What is it? I want to catch the word here.
Skeleton.
Skeleton. You have a skeleton. I have a skeleton. Bone skeleton. This skeleton is giving us, what is it?
Support.
Support. Further, it is helping us to maintain a shape. Any skeleton, if you take the word skeleton, any skeleton should have these functions. Cytoskeleton support the structure of the cell. Cytoskeleton maintain the shape of the cell. That's why it is cytoskeleton. Cyto means cell. Cell's skeleton. Clear? Are you clear?
Yes, sir.
Good. Remember, if you take a plant cell, in a plant cell, cell wall can be noticed. Cell wall gives support. Cell wall maintains the shape. Cell wall does both of them. Understood?
Yes.
Understood? Cell wall does both of them. So, if you take a plant cell, right, need of cytoskeleton is less or more?
Yes.
Less. Because there is another one to do that. Correct?
Yes.
Agreed?
Yes.
Right. But in the animal cells, no cell wall to maintain their shape, to support their structure. What is important?
Cytoskeleton.
Cytoskeleton is important. Are you clear?
Yeah.
Yes. In the cytoskeleton, my children, there are three components. The first given picture, it is called microtubule. Whose diagram is this? Whose diagram is this? Microtubule.
Tubule.
Second is this, actin filament. Third, it is this, intermediate filaments. What are the components making the cytoskeleton?
Microtubules, actin, intermediate filaments.
Intermediate filament. In that case, your book is telling cytoskeleton is made of two major components. One component is home,
Microtubules.
microtubule. Other components is home.
Protein filament.
In these protein filaments, those two filaments are included. What are the two filaments included?
Actin filament and intermediate
Actin filament and intermediate filament are included. Understood?
Yes, sir.
Right. You can see the microtubule and actin filament. They are spherical ball structures. Can you see?
Yes, sir.
They are called protein subunits. What is the common name? What is the common name? Protein subunits. Remember, in a place where a microtubule is needed, the protein subunits will assemble and form the microtubule. After its need is over, they will again detach. Now tell me, is microtubule a permanent one?
No.
No. Is actin filament a permanent one?
Yes.
No. Actin filament is not permanent. Microtubule is not permanent. Normally, intermediate filaments are permanent. Understood?
Yes.
Right. But anyway, your book is telling a common idea. Your book says cytoskeletal components can break, separate, and again form, again separate, again form, again separate, again form. This kind of a nature is there. That nature is described by which word?
Dynamic.
Dynamic. Clear? Clear?
Yes, sir.
Good. In the cytoskeleton, okay, three components there, you know, and what is their function? Providing
Strength to the
strength to the cytoplasm. That is a skeleton's function, correct? Yes or no?
Yes, sir.
Yes. What is the second function you appointed? Read the second function, Buddha.
Anchorage organelles and cytosolic.
Encouraging organelles. Now, can you see this diagram?
Yes, sir.
Okay. Can you see the blue color large material in the middle of this cell?
Yes.
Yes. What is it? What is it? Nucleus. Now this nucleus, it's fixed in the middle. By whom? By whom? Cytoskeleton. Nucleus can't go to this side or that side. It should stay in its position. Clear? Clear?
Yes, sir.
Yes. This kind organelles are placed in their places. Like, okay, like they wish they can't move here and there. Okay, that is what they are telling the word. What is it? Encourage. What are the components encouraged?
Cytosolic enzymes of the cell.
Cytosolic enzymes of the cells are encouraged. Organelles are also encouraged. Correct? So
Yes.
nucleus is encouraged, means it's an organelle encouraged. Are you clear?
Clear.
Right. Next it is movement of, what is it? Movement of cytoplasm. Second one, what is it? Read this wording.
Cytoplasmic streaming.
Cytoplasmic streaming. Remember, cytoplasm can be made to move. Cytoplasmic, what is it? Streaming. I will show you a cytoplasmic streaming. This is fine. Can you see the cytoplasm is moving?
Yes, sir.
When there are chloroplasts like certain organelles, they also will move along with the cytoplasm. That is called cytoplasmic streaming. Okay, the force needed for this movement is given by whom? Cytoskeleton. Third one,
Positioned organelle.
positioned organelles. Movement of, you have to read like this. Movement of positioned organelles. Whose movement?
Positioned organelle.
Position organelles. You include that word. Movement of whom? Movement of
Positioned organelle.
positioned organelles. And finally, movement of chromosomes. Understood? Who are the components made to move? Cytoplasm is made to move. Organelles are made to move. Chromosomes are made to move with the contribution of
Cytoskeleton.
cytoskeleton. Last one, maintaining the shape of the cell. Right. Are you clear? Are you clear?
Yes, sir.
Right. Now a table is given to you, a small detail is given to you. If you take the microtubule, what is it?
Microtubule.
Microtubule, first of all, microtubule. Right. Right. I am sharing this picture to you for microtubule. If you take the microtubule, it is made of globular protein units called tubulin protein. What kind of protein? What kind of protein? What kind of protein?
Tubulin.
Tubulin protein. Here count and see how many tubulin proteins are making a ring. Count and see.
12.
How many?
13.
13. How many are there? How many are there? 13. Starting from every one, columns are made. This is a column of, this is a column. This is another column. This is another column. This is another column. Like this, how many columns are there? How many columns are there?
13.
13 columns are there. In the center, you will not find anything. It is hollow. Clear? Are
Yes.
you clear? Good. So that is regarding the microtubule. Okay. Now we speak about whom? Microfilament. Another name for microfilament is actin filament. In the actin filament or microfilament, there will be two chains. Can you see the two chains? Can you see
Yes.
the two chains? The two chains are twined. The two chains are, what is it?
Twined.
Twined. Turn, and it is twined. Understood?
Yes, sir.
The two chains are twined. Right. Here also, the protein subunits are globular. Read and see this. Read and see this. Try to understand the two contents before I discuss the intermediate filament.
Done, sir.
Okay.
Okay, sir.
The third type of protein, it is intermediate filament. Right. So remember, microtubule, actin filaments, both are globular proteins, but intermediate filaments are made of fibrous proteins. What kind of proteins? What kind of proteins?
Fibrous protein.
Fibrous proteins. They will be now like a rope. Every protein unit will be like a rope. They will be tightly coiled around. Okay, right. So your book is using the word cold. They are super coil. Ayyo. Right. So your book is telling the word they are fibrous proteins, super coiled, tightly coiled, thicker cables. And there are many different types of intermediate filament proteins. but the full syllabus is based on which protein?
Keratin.
Keratin protein. Clear?
Clear, sir.
Right. Right. That is regarding the cytoskeleton. After cytoskeleton, we are coming to the heading called which units, my children?
Cilia and flagella.
Cilia and flagella. Okay. If you take cilia and flagella are locomotory structures. What kind of structures? Locomotory structures. Now, I want to share this one with you. You know euglena, correct? Do you know euglena? Do you know euglena?
Yes, sir.
Yes. So, when euglena, you see this picture, you see this video, the movement of euglena. This is how the euglena is moving. When it is moving, it is beating its flagella. Can you see? Can you see?
Yes, sir.
How many flagella are there?
2.
2 flagella. Helping the any organism to move. Locomotor structure. Clear?
Clear, sir.
Yes. Similarly, if you take paramecium, you can see tiny hairs. Okay, can you see that? Have you seen that one?
Yes, sir.
Okay, so we will continue this one from the coming class, inshallah. Okay,
Okay, sir.
we'll meet inshallah. Assalamu alaikum.